So since we're currently in the second block of the course, which is all about enzymes, we're going to look at the structures of a few different enzymes in this protein structure activity. So the first enzyme we're going to look at is phosphofructokinase. It's technically phosphofructokinase 1, but you don't really need to worry about. So you will meet this enzyme in the fourth block of this course when we start talking about metabolism. So phosphofructokinase is the main regulatory enzyme in glycolysis. And so glycolysis is a 10-step process where glucose, right here, is converted by a series of enzymatic steps, resulting in pyruvate, which is this molecule here. So actually 2 molecules of pyruvate for each of glucose molecules. So that's glycolysis. So phosphofructokinase is located right here in the glycolysis pathway. So it catalyzes the conversion of fructose 6-phosphate into fructose 1,6-bisphosphate. So another form of the reaction is shown right here. So fructose 6-phosphate is a carbohydrate. It's just a fructose with a phosphate on the 6th position of the carbohydrates. And then phosphofructokinase, using a phosphate from ATP, adds another phosphate onto fructose 6-phosphate to get fructose 1,6-bisphosphate. So then there's 2 phosphates, one at the 6th position and one at the 1 position. So as I said, it's the main regulatory enzyme in glycolysis. So it's actually allosterically activated and inhibited, which we'll talk about in lecture 9, I believe, 9 or 10. So it's allosterically activated by AMP. Sorry, this should be AMP. And it's allosterically inhibited by ATP. And so that makes perfect sense. So when you're breaking down glucose, you're doing that to ultimately produce ATP for the most part. So if there's a lot of ATP already, phosphofructokinase will be shut off. So you won't break down more glucose to make more ATP. If there's a lot of AMP in the cell, that means that you need to convert that into ATP to give energy. So AMP will actually activate phosphofructokinase. The other feature of it is that it requires a magnesium cofactor, and it's a homotetramer. So that ties into our other lessons about cofactors. Metal ions are often cofactors in enzymes, and this enzyme also is a homotetramer. So that's phosphofructokinase. And so the structure we're going to be looking at, it has the 4-letter PDB code 1PFK. And so it's phosphofructokinase from E. coli. So E. coli is capable of doing glycolysis as well, of course. And so it was deposited a long time ago in 1989. It's a very important enzyme, so that's not very surprising. And it was determined by X-ray diffraction at a resolution of 2.4 angstroms, which is quite good. And so here's the structure of phosphofructokinase, and you can see it's actually made of 4 different subunits. So you can see the substrates in each of the 4 active sites, which is colored a different color. You can see that it's made both of alpha helices and beta sheets, which you can see in the internal environment of each of these subdomains. And now we can focus in on one of these active sites here, and you can see that there's 2 substrates and a cofactor in this active site here. So we have an ADP molecule right here. So this would be after what the substrate would look like after the reaction took place, because that 3rd phosphate is going to be transferred to the substrate. And the substrate here is fructose 1,6-bisphosphate. So you can see a phosphate molecule in orange on one side and another phosphate molecule in orange on the other side. And then in the middle here, this green sphere is a magnesium ion that's necessary for its catalytic activity. Now, if we look at the surface of this enzyme, you can actually see how it gives you a different view of that active site. You can see that there's a really good cleft there, and that those substrates fit perfectly within that cleft. So that gives you an idea of how enzymes have specificity. So these substrates fit just perfectly into the way that this active site is structured. So if we look at the individual amino acids involved in forming this active site and interacting with the substrates, you can see that there's many, many different residues that are involved in interacting with all these specific substrates. An additional thing is that the water molecules are also present in this active site, and they can coordinate, and so they're shown here as these red spheres, and they can coordinate with different amino acids and with the substrate itself, because of course water molecules have polarity, partial positive charges with the hydrogens and partial negative charges on the oxygens. And this program actually allows us to look at the connections between these different residues with the substrate and with the water molecules. Now, if we zoom into this magnesium ion here, you can see how it's coordinated into that active site. So that magnesium is going to have a 2+ positive charge, and so that's going to interact with these different groups. So it interacts with the negatively charged phosphates on ADP and on fructose 1,6-bisphosphate. It interacts with the partial negative charges on these water molecules above and below, and it interacts with the negative charge on this aspartate amino acid shown right here. So it's basically every single angle around that magnesium ion. The magnesium ion is neutralizing or coordinating with those negative charges. Now we can also look at a different interaction. So on the other side, on this other phosphate molecule involved with fructose 1,6-bisphosphate, and you can see that the negative charge over here on the phosphate molecule is interacts with the positive charge on this arginine residue right here and this histidine residue right here. And you can see the somewhat hydrophobic body of the carbohydrate, which is shown in magenta, is sort of interacting with the surface at this methionine residue, which is a hydrophobic residue that's located right there and sort of gives it a shelf or a surface on which to interact. And lastly, we can look at this ADP molecule, and you can see in the region on the left, there's an arginine residue that has a positive charge, and that interacts with a water that then interacts with the phosphate molecule. So that's going to coordinate those. There's also a water molecule associated with one of these hydroxyl groups on the ribose sugar of this ADP molecule. And then there's another group of positively charged residues over here. So there's I believe three arginine residues that all have a positive charge and are coordinating with water to sort of interact with that purine ring on that ADP molecule. So overall, we can see that this really important enzyme, phosphofructokinase, you can see how enzymes are designed to interact with their substrate or substrates very specifically. And that's also the case with this enzyme here. We saw that the surface and the specific amino acid residues that interact with these groups, have evolved perfectly to interact with its specific substrate so that it can do its catalytic activity. Now, the next enzyme we're going to look at is also another really important metabolic enzyme called citrate synthase. So it catalyzes the first reaction in the citric acid cycle. So we just talked about glycolysis in terms of phosphofructokinase. That results in the production of pyruvate. And that pyruvate is then converted by another enzyme into acetyl-CoA. And acetyl-CoA will enter what's called the citric acid cycle, which is a metabolic cycle. So the last product, oxaloacetate, will actually be used in the first step of the cycle. It'll be combined with acetyl-CoA to form citrate. So citrate synthase is the enzyme that catalyzes this specific reaction. So it combines oxaloacetate and the carbons from acetyl-CoA to form the 6-carbon molecule citrate. So we have a 4-carbon molecule oxaloacetate here. We have these 2 carbons in acetyl-CoA right here, and they'll be combined in a condensation reaction, which is an enzymatic reaction involving the use of water to form the 6-carbon molecule citrate. And then that'll be used for the next step in the citric acid cycle. So citrate synthase is one of the main regulatory enzymes in the citric acid cycle. There's a couple that are under allosteric regulation. So it's active when just like phosphofructokinase, it's active when ATP is needed, and it's inactive when ATP is abundant. So citrate synthase in humans has 2 identical subunits, which you can kind of see here. There's 1, 2, 1, 2. And it has it's a really good example of the induced fit model when we're talking about enzymes. So when it's actually bound to its product, like citrate, so that's right here. So this is the form bound to citrate. It's actually in an open conformation. You can see there's sort of an open area right here. But when it closes around its substrate. So when it's bound to acetyl-CoA, as you can see here in green, it forms a more closed conformation. We'll take a closer look at that in a minute. So we'll actually look at 2 different conformations of citrate synthase, the open and the closed form. So one is bound to citrate, this open one, and one is bound to actually citrate and acetyl-CoA, and that makes the closed form. So the closed form, its code is 2CTS, and the other one is 1CTS. And so these were also deposited a long time ago, the year I was born in 84, and that's probably why they got such good codes. So CTS for citrate synthase. Nowadays, you just get a random code if you deposit a protein structure. And so both of these are actually porcine citrate synthase. So they're from Sus scrofa, which is a pig. And they were both determined by X-ray diffraction. The closed form is actually a little bit higher resolution. It's 2 angstroms as opposed to 2.7 angstroms for the open conformation. And that actually is pretty common in crystallography. If you have a substrate that makes the protein very stable and in a really strong conformation, that will that sometimes will increase the resolution. So this one is actually bound to CoA, not acetyl-CoA. The acetyl part would be transferred onto oxaloacetate to make citrate. So we'll take a look at this now. So this enzyme here is citrate synthase, and it's interacting with, or it's bound to a citrate molecule, which is the product of the reaction that it catalyzes. So it catalyzes the conversion of oxaloacetate and acetyl-CoA into citrate. And so this is in the open conformation, and you can see the citrate molecule here in orange, and then it's next to this very open cleft right here, and there's another one right here. So this is actually a dimer, a homodimer, and so this is in the open conformation. Now again, if we zoom in, you can see that citrate molecule is perfectly accommodated within the surface of the area where it binds in the active site of the enzyme. If we zoom in and look at the residues that are interacting with citrate, you can see that there's a lot of different positively charged residues, so histidines, a lysine, that can interact with the negatively charged carboxylic acid groups on this citrate molecule that's shown here in orange. There's also like a phenylalanine located right here that can interact with the carbon chains on that citrate molecule that are non-polar. So if we switch now to the closed conformation, this structure is actually bound to coenzyme A and to citrate. So this would be basically acetyl-CoA after the reaction has occurred, but before the CoA cofactor is released. And so this is in a closed conformation. You can see the CoA molecule shown right in the corner here in magenta or orange. And then now that big cleft is gone. So that's an induced fit in this citrate synthase molecule. When it binds to the coenzyme A, it forms this closed conformation. And again, if we zoom in, you can see that substrate fits within this pocket really nicely in that closed conformation of citrate. You can also see again that there's a lot of positively charged residues interacting with the polar negatively charged groups on this coenzyme A molecule. So here you can see how this molecule is able to switch very easily between this open and closed conformation. So we talked a lot about proteases in the section on enzymes. So we have, trypsin and chymotrypsin and thrombin. Another one is called pepsin. So pepsin is a protease that's found in gastric juices, so in your stomach. And so what it'll do is it'll cleave polypeptides that you basically ingest. So if you have some chicken or steak or some beans and they have proteins in them, pepsin's one of the peptides that will cleave those proteins to release the amino acids so you could use those to make proteins in your own body or use them for energy. And so pepsin preferentially cleaves after leucine, tyrosine, tryptophan, or phenylalanine residues, so larger aromatic or hydrophobic residues. So it was the second enzyme after urease to have its structure determined way back in the 1930s. And that was a really important research because it confirmed that enzymatic activity was caused by proteins. And so here's a picture of the protein crystals actually from way back when that were made of pepsin in the 1930s. Now, we're just going to look at one feature of pepsin. So here's the structure of pepsin. We'll look at it a little bit closer in a second. But to avoid proteolytic activity of pepsin in the cell, it's actually synthesized as something called a zymogen. And this is an inactive sort of pre-processed form of a protein or an enzyme. And that's called pepsinogen. And so that's right here. And so this actually is synthesized with this protein. Part of the protein is actually embedded inside the active site cleft. So before it gets to your stomach, while it's being made in the cells, it won't break down proteins within the cells. So once it's in the stomach and it's supposed to be doing the job that it's supposed to be doing, that part of the amino acid will get cleaved off. And you have this nice, long, clear active site right here that can interact with polypeptide chains. And so we'll be looking at the two different forms of pepsin. So this one, 3-PSG, is pepsinogen, and this one, 5-PEP, is pepsin. And so these structures were both deposited in the 90s, and these are actually both porcine pepsin as well. They might have actually been isolated from the gastric juices of a pig. So that was one of the reasons why it was studied so early. You could get a lot of pepsin just from stomach acid before you could produce proteins through recombinant methods. And so these were both determined by X-ray diffraction. Pepsinogen has a really high resolution of 1.65 angstroms, whereas pepsin also has a decent resolution of 2.34 angstroms. And so now we can take a quick look at pepsin. So this is pepsin right here. I'm just going to show the surface of this pepsin molecule in pink. And so this is the active form of the enzyme and its catalytic site. So the actual residues that are going to perform catalysis that are going to break those peptide bonds, those polypeptide bonds that pepsin interacts with, are shown in red. So you can see that there's a really deep active site around that catalytic site right there in pepsin. So when pepsin is first synthesized, it's synthesized as a zymogen, so a pre-enzyme or a pre-protein molecule that needs to be modified before it has activity. So you can see this green portion right there is also part of the protein, but it blocks that deep active site in pepsin. So this zymogen is called pepsinogen, and it has no activity. So when it's synthesized in the cell, it's not that pepsin molecule is not going to immediately chew up everything in the cell like it is sort of designed to do or supposed to do or has evolved to do. And so when the pepsinogen is inactive until it's released into the gastric, into your stomach, basically, and then that green part will be cleaved off by another peptide or by another protease, and then the pepsin molecule will have full activity. So the next enzyme we're going to look at is oxidosqualene cyclase. So this enzyme is actually involved in the synthesis of cholesterol. So cholesterol is a very important lipid molecule. It's involved in regulating cell membrane fluidity, and it's also the precursor for the synthesis of vitamin D and other steroids. So cholesterol is really, really important. So it's a steroid as well. I don't think I've mentioned that before. So it's a steroid, and that means it has these 4 fused rings, as you can see here. So this is cholesterol right here. It's got these 4 fused rings, but it's still a lipid molecule because it's primarily just made out of hydrocarbons. So The process of making cholesterol is actually really complex. There's like 24 enzymes that are required for cholesterol synthesis from precursors. And oxidosqualene cyclase actually performs one of the more complicated reactions. So what it does is it takes this hydrocarbon squalene, or actually a modified version of it, oxidosqualene, and then oxidosqualene cyclase will convert that molecule, and I'll actually take what is basically a long hydrocarbon ring, and it'll condense it into form those 4 fused rings. So that's a really important reaction for your body. So because this will make the molecule lanosterol, which will then be converted into cholesterol, and then cholesterol will do its job and then also be converted into things, other vitamin D and also other steroids like cortisol, testosterone, estradiol, stuff like that. And so this oxidosqualene cyclase is actually associated with microsomal membranes. So it'll access these squalene molecules, or these oxidosqualene molecules, directly from the membranes and then put the lanosterol back into those membranes. So here's the structure, like a cartoon of the structure we're going to look at. So it's actually bound to lanosterol. So that's right here. And it actually has this region here that has these lipid molecules bound to it. And so that's where it would be associated with the membrane. And so here is the structure of oxidosqualene cyclase we're going to look at. So its code is 1W6K. And it's complexed with lanosterol. It is the oxidosqualene cyclase from Homo sapiens, from human beings, but it was expressed in yeast cells, actually. I think this is the first recombinant protein that we've looked at that was expressed in yeast. So you can make other proteins in yeast recombinantly. So it was deposited in 2004. X-ray diffraction, it has a very high resolution of 2.1 angstroms. And so now we can take a look at oxidosqualene cyclase, which is shown here in yellow. So it's actually bound in the middle there to a lanosterol molecule shown in blue, and then several lipids on the side here shown in green. So if we take a look at the surface of this molecule, you can see that you can't even see that lanosterol molecule in the middle of the protein. So it's actually buried in the interior of the protein in this conformation. You can still see the lipids on the right-hand side, but that lanosterol molecule is buried within the protein. So if we take a closer look, you can actually see that there's a bunch of different, so lanosterol is a hydrophobic molecule for the most part. And so there's a bunch of different hydrophobic residues that are interacting with this lanosterol molecule. So there's tryptophans and a valine right here and a leucine and stuff like that. So these hydrophobic residues are actually going to interact with the actual lanosterol molecule and with the oxidosqualene molecule before it forms lanosterol. And it's the same thing over here on the side. So this is going to be the part of the protein that actually interacts with the membrane. And so this crystal structure was actually able to capture some hydrophobic type, lipid type molecules in this region. And you can see this part of the protein that interacts with the lipid hydrophobic membrane contains also a bunch of hydrophobic molecules, which are shown in dark gray. And so that's oxidosqualene cyclase, very interesting enzyme that acts on a hydrophobic molecule to form, it's very important to help form cholesterol and other steroids. So xanthine dehydrogenase is a pretty cool enzyme too. So what xanthine dehydrogenase does is it helps break down extra purine nucleotides. So that's ATP and GTP from either your diet or just from your cell if you have excess amounts of ATP or GTP. And it does this by this reaction here. So it converts these ATPs or GTPs into uric acid or urate, which is shown right here. And so urate can actually be excreted and is a normal component of urine. That's how it gets the name. And so xanthine dehydrogenase does that. So you'll come across a lot of dehydrogenases. Those mean that there's going to be an oxidation reduction reaction. So that we'll talk about it later in the course, but you can just use the device oil rig. So oxidation is loss of electrons, and reduction is gain of electrons. So in these dehydrogenases will these are enzymes that catalyze these oxidation reduction reactions, where one molecule will lose electrons and then another molecule will gain electrons. So in this reaction, the purine nucleotide is actually oxidized to urate. So this is oxidized. Cannot write well today. And then this molecule, NAD, which is another cofactor we'll talk about a lot, is reduced. So this is reduced to NADH. So it'll gain a hydrogen atom and electrons as well. So how xanthine dehydrogenase does this is it transfers these electrons through the enzyme. We'll take a look at that. That's basically what we'll focus on, looking at the enzyme. And so it'll take from this molecule, this purine molecule here, and those electrons will be transferred to a molybdenum ion, which you need molybdenum. It's just an element. You need it in trace amounts, partially for the activity of this enzyme, and then it'll transfer those electrons through these things called iron sulfur clusters, which are sulfur molecules bound to iron, and then it'll transfer it to NAD to make NADH. So high levels of uric acid buildup from this activity from xanthine dehydrogenase is actually involved in diabetes, kidney stones, and gout. So if you have too much rich food, stuff like that, too much uric acid can actually cause a lot of problems. And so here is the structure we're going to be looking at, xanthine dehydrogenase. And so it's 1FO4, and it was actually isolated from cow milk. So a lot of times you can just take milk, and there's a lot of this enzyme happens to be in milk. So the organism it's from is Bos taurus, which is a cow. It was released in 2000. Also X-ray diffraction, and also a very high resolution of 2.1 angstroms. So now we can take a look at xanthine dehydrogenase. I apologize that it changed to a black background. My animations were getting a little choppy for some reason. I'll try to fix that. But this is a very large protein. It's actually made of two, it's a homodimer, so two subunits of the same polypeptide chain. And you can see there's alpha helices and beta sheets throughout this protein. So xanthine dehydrogenase is a dehydrogenase enzyme, of course. So it's going to transfer electrons from a purine molecule and ultimately transfer those to an NAD molecule to form NADH. So that's basically what happens with dehydrogenases. So how are those electrons transferred from the substrate to the co-substrate to form the product NADH? Well, here's a molecule in here that is a purine molecule. And so the electrons are going to be basically stripped off of this molecule, and they're first going to be transferred to this molybdenum molecule that's shown next to it with these yellow, red, and green molecules right here. So the molybdenum is actually shown in green, and it's coordinated with a couple different atoms. And so those electrons from the molybdenum are actually going to be transferred to this group, which is an iron sulfur cluster. So we have the yellow, we have sulfur, and then in this orange is going to be iron. So those molecules are actually able to accept those electrons that came from the purine, went to the molybdenum, and then were transferred to this iron sulfur cluster. And then that iron sulfur cluster will transfer those electrons to another iron sulfur cluster a little bit away on the protein. And then lastly, those electrons from that second iron sulfur cluster will then be transferred to, actually this is FAD in this molecule here, but it would either be FAD or NAD+. So it would transfer the molecules to that molecule, and FAD would be reduced to FADH2, or NAD+ would be reduced to NADH. So we'll talk a lot about FAD and NADH and oxidation reduction reactions when we talk about metabolism, because they're really important in the production of ATP. And the last enzyme that we're going to look at just very quickly, because I mentioned it a few times in the lectures, is carbonic anhydrase. So this is about, I think, the fastest enzyme known. So it's really important in your body. It's in every single different, organism on the planet. And so it converts CO2 and water to form carbonic acid or bicarbonate and protons. So it's present in almost all organisms, including plants and algae. So in plants and algae, it'll actually convert this bicarbonate back to CO2 for respiration. And in corals, so it's actually involved in forming calcium carbonate in coral polyps. So it does require a zinc ion in every single form. So there's a few different forms, like in humans versus plants and stuff, but it requires a zinc ion in all those forms. So that's why you need zinc in trace amounts. And it can be inhibited by drugs like dorzolamide, which is shown here, and is in the structure we're going to look at. So we'll just look at a dorzolamide bound carbonic anhydrase. So this molecule is actually used because it's involved in the release of these protons, that it has a lot of effect on like fluid buildup in the eyes. So glaucoma is caused by excess fluid in the eyes, increasing the pressure in the eyes. And so dorzolamide is one of the first drugs you would give to treat glaucoma. So my wife's actually a veterinary ophthalmologist, so she gives dogs dorzolamide quite often. And so here's the structure we're going to look at very quickly. So it's 4M2U, carbonic anhydrase in complex with dorzolamide. The organism, it's from humans, but it was expressed in E. coli. So this is a recombinant protein that was purified. And it was deposited in 2013, determined by X-ray diffraction with a high resolution of 2 angstroms. And so lastly, we'll just take a quick look at carbonic anhydrase because we kept talking about it so much in the lectures. It's the fastest enzyme known for the most part. And so here is the structure of that molecule here. You can see it's made up of an internal beta sheet region surrounded by a few loops and alpha helices. I'm not going to go into detail about this, but in yellow is actually that inhibitor dorzolamide of carbonic anhydrase, and you can see how it fits really well into the active site of carbonic anhydrase, which makes it a really useful competitive inhibitor. So it's going to compete for that active site with its natural substrate. So that sphere in the middle is a zinc ion, and you can see how it's coordinating with both the dorzolamide and these two histidine molecules that are going to coordinate with that zinc ion. And so just take a minute, pause it, and just take a look at some of the amino acids that are surrounding this active site of this very, very important and medically important enzyme.